Welcome. My name is Kristiina Aasa. I'm the product manager here at FUJIFILM VisualSonics, based in our Toronto headquarters. And I'm very happy to be moderating our session today and welcoming you and all of our internal experts here to ask some questions about photoacoustic imaging. So to get started, I just want to run you through how the session will go today. If you've attended our webinars in the past, this will be quite familiar for you. We are recording this session for today, so if anyone wasn't able to join or you wanted to rewatch some of the answers to your questions, that will be possible. Do expect the webinar to run about 45 minutes or so. Everyone's lines are muted for the duration of the webinar, but please use the Q&A button in your Zoom panel at any time in the session, including right now, to start entering the questions you have about photoacoustics so we can queue those up for all of our experts to answer. So to get started, before I pass things over to all of our experts, I wanted to make sure everyone's aware of our new flagship products. We've had some changes in the last year and a half or so, so I want to make sure we introduce the Vevo F2XC, which is our new ultra high frequency ultrasound system, which is pictured on the screen here, as well as our new Laser X20, which is our flagship photoacoustic laser. So this was launched, the XC was just launched actually about a month ago, and our Vevo Laser X was launched about a year ago. X20 was launched about a year ago. So they're both fairly new, and together they unlock the capabilities of ultra high frequency ultrasound and photoacoustic imaging for preclinical use. So a little bit about our brand new Vevo F2XC. It is our flagship platform. It's the next version of the Vevo F2 with extra channels and extra capabilities in order to exceed your expectations. The Vevo F2XC is our high frequency ultrasound platform. It pairs with our Laser X20 to deliver comprehensive multimodal data and propel biomedical research forward in the investigation of preclinical disease. It has a broad frequency range of 1 to 71 megahertz, which offers versatility to be able to image any animal or disease model. We offer high throughput scanning with higher or faster acquisition of photoacoustic imaging, faster 3D and 4D reconstruction, and on the analysis side, AI-assisted analysis. We offer some advanced capabilities and advanced imaging on the back end to enable more complex data sets to be acquired. It is proven and validated technology. Hopefully many of you are already using our technology or have been for many years. So we are well validated across a wide range of application areas, including cardiology, oncology, neurobiology, and many, many more. And we'll hear about that and everyone's specific expertises in a few minutes. And ultimately, it gives you the image quality and resolution you need to quantify the most subtle indicators of disease in your models. So when we talk about photoacoustics and adding photoacoustics to our ultrasound, we really talk about adding another layer of data to do multimodal imaging and advanced tissue characterization. So a few things about our Laser X20, if you're not familiar with it. The Laser X20 is, again, our flagship photoacoustic platform that offers advanced multimodal imaging. You're able to see endogenous and exogenous agents in the body with an expanded wavelength range of 660 to 1320 nanometers. We're able to choose your energy level that suits your needs for your research. So this is a high energy laser, and we'll talk about that in a minute, but you are able to actually attenuate the energy down to whatever your research dictates. Combined with the Vevo F2XC, we're able to visualize data with double the speed and enhanced image quality for our photoacoustics. On the analysis side, we're able to do more accurate detection of in vivo spectral shifts using our spectral unmixing software. So a little bit more about the value of that longer wavelength range. So the wavelength range has changed depending on which laser you're familiar with. So for our new X20 laser, that is 660 to 1320. So when we talk about the added value of that range, we're talking more in that higher end, highlighted in yellow here. So we can see, of course, we can still detect oxygenated and deoxygenated hemoglobin. And you can see the curves indicated here, as well as melanin. But now we can investigate and interrogate these additional curves here. So in orange, we have lipid, which peaks at about 1210 nanometers. And then we also have this theoretical collagen curve in purple. We call it theoretical because it's basically, we know there's many different subtypes of collagen, and this is just um, an amalgamation of those different curves. So it's more on the theoretical end. But what that lets you do is visualize things like lipids in the body. So this is an image of the posterior of a mouse. So we can see the fat on the surface of the skin in yellow here as lipids, and then extending down around the back muscles, which is certainly what we expect. We can see that in a disease model here, or an aged model, where we have a normal mouse on the left-hand side and an aged mouse on the right. What you can see in green is that lipid signal. So in the healthy animal, we just see that layer of lipid underneath the skin, which is quite normal. But in the aged mouse on the right, we can see that extended area of lipid under the skin, as well as some presence of lipid within the liver. When we talk about the value of flexibility and energy levels, we can see here's the energy curve for our laser X20. You can see the maximum energy from that laser is quite high, peaking at around 100, just over 130 millijoules. And then it kind of decreases as the wavelength increases, which is expected. But as I mentioned, you can attenuate that energy if you don't want to utilize it all. So we can go to a high level of energy, which caps that energy level at 80 millijoules. You can go to a medium energy level, which caps it at 60, or you can go even to a low energy level, which caps it at 40. So this is really great for people using sensitive nanoparticles or agents that might be prone to melting at high levels of energies, let's say. So really our system offers, and we can certainly, I could have a 2 hour, 3 hour lecture on this, but I wanted to keep it very short so we can get to your questions and really bring in our experts to talk. But the Vevo multimodal imaging system offers you methods to do anatomical analysis, so detection of pathology, 2D and 3D scanning and quantification, functional imaging, so you can do, of course, cardiac function, vascularity, perfusion, even functional brain imaging, molecular imaging. So you can, this is where the photoacoustics really comes into play, where you're looking at oxygen saturation, let's say in a tumor, or the tumor microenvironment, biodistribution of targeted agents, let's say. And then mechanical, so new on the F2, and it's been around for about 2 years now, is the ability to do shear wave elastography. So we can actually get mechanical information about tissue non-invasively using our system to do things like fibrosis detection in the liver. So with that, I would like to invite my colleagues to join me on screen here. Everyone can turn on their cameras, and we'll introduce you to the team here. So just in order of our kind of my order I can see on my Zoom. So Drew, if you wouldn't mind introducing yourself.
Sure, thanks, Kristiina. Hi, I'm Drew Heinmiller. I'm the acting director of product management here at FUJIFILM VisualSonics, but today I'm kind of tapping into my past experience at VSI as formerly an apps person specializing in photoacoustics, helping to develop and bring to market the first commercially available photoacoustic system way back when, affectionately referred to as the blue laser, and subsequently being the product manager managing those products. So happy to be here.
All right, next up we have Dylan.
Yep. Hello, everyone. I'm Dylan Lawrence. I am an application specialist, and my background is in photoacoustic imaging and developmental biology. So have worked in many different applications since joining VisualSonics, and excited to keep learning and keep seeing what we can do as we advance the technology.
Great. And maybe if everyone could also indicate where you're based. Drew, you're
Yep.
based in Toronto. Dylan, yourself.
I am in Houston, Texas.
There you go. And then finally, Peter.
Hello, good afternoon. I'm Peter Kesa. I'm application specialist at VisualSonics for almost 5 years, and I'm enjoying the high-frequency ultrasound and photoacoustic imaging. I especially like imaging of either subQ tumors or orthotopic tumors, but I'm open to answer all your questions today. So enjoy.
And Peter, where are you based out of?
I'm based in Amsterdam. Thank you.
Great. So as you can see, we have a nice variety of representing our global kind of team that is all out to help everyone. So again, I want to remind you that we do have that Q&A button. This is supposed to be more of an interactive session, so we've had some questions come in even before or during registration, so that's great. But please feel free to input any of your questions into the Q&A panel, and we will get our experts to chime in and answer them. So to get started, and whoever wants to answer these questions, please just speak up. If no one speaks up, I'll have to put someone in the hot seat and assign them. So first we'll start, what kind of information can I get from tumor scanning with photoacoustics? Don't be shy.
Peter, you mentioned you've
Yeah,
done a lot
okay,
of
yes,
tumor
thank you.
scanning.
Thank you. Maybe I can answer this question. So keep in mind that when scanning with the photoacoustics, there is always anatomical co-registration. So at the same time, let's say after the 3D scan, you will get information on the tumor volume, but added molecular information may contain information on the total hemoglobin content, for example, the average hemoglobin concentration, oxygenation. And in the case that you are interested, let's say, in tracking of some special drug carriers, labeled drug carriers, you can also get information on the biodistribution within the tumor. The 3D scanning, for example, in photoacoustics will gives you an overall information on the distribution of the hemoglobin. So by assessing the different areas in tumors, you will see more or less vital parts of the tumor, and this may result in scanning of or searching for necrotic areas within the tumor. But here I wouldn't stay only with photoacoustics because it's always necessary to combine this, for example, with either with the Doppler imaging or with a nonlinear contrast imaging. So for example, for when assessing the tumor perfusion. So using one multimodal system, you can get overall information on tumor microenvironment.
Great.
Yeah, I think
Thank you, Peter.
a lot of users will be a lot of people in oncology research will be familiar with optical systems where you're looking at bioluminescence or fluorescence, and I think this is where photoacoustics really shines. Is it is a hybrid optical ultrasound modality, so you really are getting optical signals. The difference between those purely sort of optical techniques with fluorescence and bioluminescence is that with photoacoustics, you're detecting those optical signals with ultrasound. And ultrasound penetrates deep in the tissue and you can resolve it because it doesn't scatter like light does. So you're really seeing optical signals, but they're deep in the tissue with high resolution. And especially with our ultra high frequency technology, we get down to 40 micron resolution with those optical signals. So you're really looking, instead of just looking at sort of the surface of tumors, you're looking deep into tumors, orthotopic, sub-Q, all sorts of different tumor models. And as Peter mentioned, just having all these other access to all sorts of other information about anatomy, like necrosis and blood flow with Doppler and contrast and so on.
Great. Thank you both. And I'll just add to what Peter was saying as well about oxygen saturation. So there's been several studies in oncology and investigations of new novel therapeutics where they found that assessing oxygen saturation was really valuable to predict things like tumor recurrence or responsiveness to therapy. You want to make sure there's active vasculature in that tumor because we know that tumors can be quite heterogeneous. So it can be quite informative to use the photoacoustics in order to better implement your therapeutics, let's say. Great,
I think,
thank you.
Kristiina, when speaking, for example, when speaking about the transducer, about the resolution in generally, it's also good to mention that the resolution of the photoacoustics is the same as resolution of the ultrasound. And this will allows you, especially when scanning with the high frequency transducer, to see and detect the complete microenvironment of the tumors that are, let's say, in size of 2 to 3 microliters only. So this is really, really amazing, I would say, especially when you can't see that kind of tumors by your eyes, because usually those tumors are under the skin. So
Great, thank you. We talked a bit about multimodal imaging. So of course, not all the questions have to be specific about photoacoustics, and we did have someone talking more about fibrosis in the heart, which can be evaluated in a number of different ways. So can anyone here comment on the ability to detect fibrosis tissue in the heart? Dylan, you look like you're going to say something.
Yeah, I think it is definitely possible. It's something that is a little trickier. It requires, some extra consideration just because, the mouse heartbeat is quite fast. So we need to take into account, frame rate and, acquisition speed. But, that going back to the multimodality aspect of the F2XC, we can look at, the B mode and the overall anatomy and structure of the heart, and then add in some of these other analyses such as the strain analysis. Not necessarily, it doesn't necessarily have to be limited to photoacoustics.
I think in combination with, I mean, while we're on the sort of topic of heart imaging with photoacoustics, the ability to, this is where oxygen saturation can come in, and the ability to look at oxygen saturation in the myocardium. So how well is the myocardium actually perfused? And a number of years ago, we developed a tool called PAEKV to actually, address the challenge of the high heart rate of the mouse and the slightly slower frame rate of photoacoustic imaging in order to do sort of gating, essentially, gating to the cardiac cycle in order to generate, one entire cardiac cycle, but with photoacoustic imaging. This is, as far as I understand, it's unique to our system, and again, does allow you to evaluate oxygen saturation in the myocardium itself, throughout one cardiac cycle. I'm not sure. I'm not an expert on cardiac fibrosis, so I'm not sure how that relates. But if someone out there is, you can share that with us. That'd be great.
Great, thank you. Moving along, there's a question about our technology. Have we introduced a laptop system? If so, what are the capabilities? So I can maybe handle that one. So our system, I shared in the first few slides there, is a cart-based platform. High frequency linear array technologies, which is what our transducers are, require pretty substantial hardware and electronics. So when you look at the base of our system, which is kind of like the box at the bottom of the stand, it's quite large. It takes quite a bit of electronics to power the capabilities that we offer. So we're simply not able to minimize that into a laptop system without compromising significantly on the quality of your images or your frame rate. And all these things are very important to do robust preclinical work.
And in case maybe I'm interpreting it wrong, but we have a workstation software which runs on a laptop. So you do acquisition on the system, you bring your data over to the workstation, and that work's done on a laptop if that counts. So that's a lot of our analysis features, more advanced analysis features are on our what we call Vevo Lab workstation software.
Great. And there is a question about human imaging. So our photoacoustic technology and our technology as a whole is not FDA approved. It's not approved for clinical use, so we are not able to provide any input on that in this form. So someone's asking here about different types of mice. So the question is, I want to make sure something about photoacoustic imaging. The non-invasive photoacoustics applies to white mice. But what about black mice? Is there a way to detect photoacoustic signal from a black mouse? And the example that's given here is for a carotid artery. So any input, kind of comments on imaging black mice specifically?
Yeah, I can take this one. Photoacoustic imaging definitely does work on the black 6 mice. We have done a lot of work on that in both in our office and in publications in the field. The biggest consideration is obviously going to be those melanin patches that are on the skin. Just from firsthand experience, I have found that those correspond with the fur growth cycle. So if this is something that you really want to make sure that you're getting out of the way of your area of interest, you can always, go through your animals ahead of time and shave, a week before, and hopefully that growth cycle will cycle through, and the skin will become, that nice pink, same as the white mice, over your area of interest, and eliminate any interference from those melanin patches.
Can you just describe that a little more, Dylan?
Yeah.
Are you saying like soon after shaving, you'll get more
So
pigmentation
yeah,
and
so as the fur grows back in and the follicles become active in the like active growing phase, the skin in the area develops those patches.
Hmm. Hmm.
And I think that the melanin content in generally depends on the strain, so there is more variations for black mice in generally. So it really depends on the strain.
I think too, there is a bit more of an opportunity with our new product, the Laser X20, in those longer wavelength ranges. So melanin absorbs most strongly at the shorter wavelengths, and then it's sort of got this linear decay as you go out. In those longer wavelengths, we're talking, say, maybe above 900 nanometers, you actually have much less contribution from melanin at those wavelengths. Um, so that could be that provides a bit of a window that we didn't used to have on our previous products. So 900 through to 1200, you get much less contribution from the melon, and so that's another option there.
Yes, that's correct, Drew. And this is also opening the doors, for example, for in vivo real-time as imaging of, for example, lipids or in generally theoretical collagen. So there is more applications you can do. And keep in mind that in the case that, for example, the black mice they are keeping, let's say, some kind of background, so you can still work with this and do, for example the full multimodal photoacoustic imaging.
I would go maybe even a step further while we're on this sort of topic. The way our system works as well, the light delivery is not entirely fixed. So it's possible to actually decouple the light delivery from the transducer itself. So if you're not quite getting the window you need, with the light delivery sort of more or less collinear with the transducer, you can sort of illuminate from a different angle where perhaps there isn't quite as much melanin, and then receive with your ultrasound transducer. So that's another option too. It just provides some flexibility to be able to work a little more flexibly with these potentially more challenging models.
Great. Thank you all. That was very thorough answer. Awesome. So the next question is about lipid imaging and some of the differences in the approach between using multi-wavelength scans and single wavelength scans. So the question is, we've been collecting and doing multi-wavelength scans and unmixing to collect data from our area of interest. In running single wavelength scans on the same wavelengths, we found significantly higher average and average threshold values in the same area on the same animal. Is the multi-wavelength less accurate? Which one should I use for the best comparison between our animals? So really getting to the differences in how multi-wavelength scanning and unmixing works versus just using single wavelength.
Okay, if I can start answering these questions. So this is a really good question when combining, for example, two imaging modes. Let's start with the single wavelength mode. So as you know, you can just select one of the wavelength within the full wavelength range, and then you can you can do either 2 or 3D scan at this specific wavelength. But in the multi-wavelength mode, the system is automatically selecting the specific wavelengths for two or more different chromophores, either endogenous or exogenous. So it may happen that, for example, in multi-wavelength mode, the automatically selected wavelength is slightly different to wavelength you used in a single wavelength mode, for example. So this can give you slightly different PA amplitude. Always keep in mind the photoacoustic profile, the spectrum of the chromophore in generally.
Great.
Yeah, if I understand this question right, it's saying running, collecting, unmixing. So there is a very big difference between sort of unmixing and single wavelength. Single wavelength, you're firing the laser at one wavelength and you're getting a signal back. Anything that absorbs light at that wavelength will give you a signal. So you're not looking strictly at one chromophore. You're looking at all chromophores, which may have different absorption at that wavelength, but they'll all contribute to that one signal. The the advantage, I would say, of photoacoustic imaging is actually in the so-called multispectral aspects of it, where you're imaging with multiple wavelengths and you're not just looking at, what is the signal at these different wavelengths. You're doing some math in order to determine, if you know the components you're looking for, if you know the absorption profile across that wavelength range for the components you're looking for, um, you can do some math and say, okay, I'll collect a few wavelengths and I can actually work out mathematically the contribution of each chromophore at each wavelength, and then do some math and display. You're not displaying the PA signal, you're actually displaying the contribution to the signal of each separate chromophore. And this is the absolutely the power of any multispectral imaging, be it optical or any multispectral technique, is that ability to do, essentially separate components based on their absorption profile and say what they are, rather than just, oh, we have a signal at this one wavelength that's contributing something, and it's contributing less at this other wavelength. This is actually telling you, in theory, the actual concentration of the chromophore, of each chromophore that you're interested in, using a multi-wavelength approach. So they're
Yeah,
fundamentally different ideas.
yeah. I think what our advice is, and in the field when we do these trainings for the laser systems, is consistency is key. You have to figure out which approach, which acquisition type is appropriate for your study, and just make sure that you're consistently setting up your image and doing the analysis the same way every time, which will give you, the reproducible results that you're hoping for.
Great. Thank you all for that explanation and the tips there. So the next question is about back to the heart. The question about cardiac fibrosis and with photoacoustic imaging may have inferred the detection of collagen with photoacoustics. There are contrast agents for PET and MRI that target collagen, fibronectin, etc. Could a contrast agent that targets fibronectin or collagen with an ICG label be helpful for photoacoustics? Give a second for everyone to process that. So talking about whether you can use ICG as a label for things like fibronectin or collagen, and maybe kind of interrogate more the possibility of looking at cardiac fibrosis.
I think in general,
Who looks like them?
You go ahead, Peter.
Dylan. Okay, yeah. I think in general, maybe there are, let's say, commercially available different chemical modifications of the not only the ICG but also other cyanic dyes that maybe you can use to create your own specific probe, having affinity to, let's say, the fibrotic areas, and then just go for a standard photoacoustic scan and scan it.
Yeah, if I can add to that a little bit. I mean, the approach in general with photoacoustic, and specifically photoacoustic contrast agents, is to, find a particular target that's not just an endogenous chromophore. So it could be just a receptor on a cell, and design contrast agents. So these are just dyes, nanoparticles, other materials that absorb light. And ideally you want something that has a nice peak, meaning it only absorbs one color of light and not all the colors of light.
Okay.
And that means doing this multispectral imaging approach, you can unmix that particular component very easily. So it's similar to any kind of targeted imaging approach, be it optical or PET or MRI or anything else. Collagen is an interesting one because there are many different types of collagen, and it really depends on the research question you're trying to ask. I think in this case, I think we'd be interested to learn more because we have had a lot of customers asking about imaging collagen, and if there were a contrast agent that was targeted to a specific type of collagen, or this person mentions fibronectin, I think that would be really interesting, and I'd be interested to know more. There are companies out there that are developing contrast agents that have this so-called click chemistry where you really can just sort of click your chromophore onto your ligand, and target a given molecule. So if you're interested in pursuing something like this, if you've got a particular research question or target that you're after, please reach out to us, and we'd be happy to connect you with someone who might be able to answer that or even provide a solution.
Great, thank you. So staying on the topic of unmixing and contrast agents, to unmix the signal of a specific contrast agent, we need a reference curve. I think the reference curve from phantom measurements is not really reflective of a signal in vivo. Is it better to acquire the reference curve of contrast agents in ex vivo conditions? Our target in this case is the mouse brain it sounds like in a black 6 mouse.
This is, I think, a really good question because keep in mind that sometimes some type of, for example, dyes or in generally contrast agents may change their optical properties after the interaction with, for example, serum proteins or other compounds of in vivo tissue. So it's always nice when you can, in the case that you can take one spectrum in vivo and then further you can also use it for, let's say, 3D multispectral unmixing. But I think the first step is always you have to start with the characterization of your either, let's say, nanoparticle-based contrast agent or dye with a phantom, and then use this spectrum for further in vivo imaging.
If I could add on to that, I think that's a really great question. We do offer a new beta analysis software that's called blind unmixing. Basically, one of the main values of that is to look at spectral shifts that happen in vitro. So what it enables you to do is characterize your agent in vitro, and then do your in vivo scan and look for slight shifts in that curve. And then you can actually save that new curve that's just maybe a bit shifted over, up, down, but highly correlated, and save that as a new curve, say as your in vivo contrast agent, and then be able to unmix with that. We do offer that as a beta software now. And if you are interested in trying that out, please do send us a message in the chat here or contact us later. We would love to have you test that out with us.
Maybe Kristiina, is also good to mention, let's say, the new feature that is going to be available in the new Vevo app that is called Mask. So you can select the specific, as you mentioned, the specific region of interest, and assess only the photoacoustic signal from that specific region. So this may help you, and this may improve the analysis.
Yeah, absolutely, and that's certainly something we can show anyone who's interested.
Mm-hmm.
Okay, so shifting gears a little bit here, we mentioned obviously our systems are designed for preclinical use. Someone's asking, what are the main challenges to clinical translation of this technology or moving this into clinical studies?
I can maybe jump in here. There is clinical research going on with photoacoustics. There's actually a commercially available clinical photoacoustic imaging device. It's not a device we make, a different company, but there's a lot of interesting clinical research. You just do a PubMed search for, clinical photoacoustic or optoacoustics, and you'll see quite a bit of research here. Really interesting applications, everything from melanoma to, even irritable bowel syndrome, wound healing, a lot of really interesting work here. The challenge, I would say, mostly has to do with getting enough light deep enough into the tissue to be able to see something. In a mouse or a small animal, that's no problem. If you want to see, deep abdominal imaging in a human, that's a different story. The amount of light that you've got to get in order to generate that photoacoustic signal is fairly significant. And of course, light just scatters, as soon as it, gets about a millimeter beyond the surface of the tissue, it scatters and goes everywhere, and you get this exponential decay of that light energy. So I'd say that's probably the main challenge. But that needn't be a limitation to clinical applications. They would just have to have, there's plenty of, say, minimally invasive devices for, catheterization and, intravascular imaging and that kind of thing. So it's really about picking the application where you can deliver light in a way. And the interesting part is you don't need to do both the light and the ultrasound, say inside the patient. You just need to get the light in there, and you could detect the ultrasound from outside the patient with clinical ultrasound frequencies, that kind of thing. So really, really interesting work going on there. And it will be interesting to see how this progresses as well, because there are applications even in, surgical guidance and those kinds of things.
Great, thank you. Shifting gears a little bit, and again, talking about the multimodal and complementary technologies we can apply. Question about if we're doing elastography, if so, how are we specifically doing it? So we do offer shear wave elastography that was introduced on our Vevo F2 platform about a year and a half ago. So it is a plane wave implementation of elastography to create the shear wave using the same transducer you image with. So this was optimized for the liver to look at liver stiffness, for instance. And then we do have other researchers who are investigating the application of shear wave elastography using our technology in other models, such as tumor models. Happy to connect with you further and give you some more specific information about that as you require. So next question here. How fast can your instrument, and this is photoacoustic instrument, switch between wavelengths, and how fast can the instrument switch between 2D slice planes for multi-slice imaging? This pertains to dynamic scans that rapidly track agents in multiple image slices. So who wants to tackle this one?
I feel like this is an applications question. Dylan, do you want to go for it?
Sure. So the frame rate for the laser X20 it's a 20 hertz laser, so we're getting up to 20 frames per second. So it is a much faster laser than or acquisition than what we were using in the past. But switching between wavelengths, I know it acquires with the F2XC, requires 2 laser pulses per wavelength and then switches. So do the math times however many wavelengths in your 20 hertz repetition rate, and you can figure out the speed for the specific application. In terms of switching between 2D and 3D imaging, that also depends on the scan size and the step size. For the multi-wavelength scanning, our system does take every wavelength at each position before it moves on to the next. So again, there's hard to give a specific number because there's a lot of variables that can come into play.
I've actually heard this asked before, and it's an interesting one, and perhaps something we need to talk to our development team about. Yeah, so the wavelength switching that's easy. It switches between pulses, so you can go from I think the whole range, from 660 all the way up to 13 something between pulses. So you can do that, that's no problem, is switching. I think the bigger issue is, sounds like this person wants to go between, say, 2 imaging planes. So I want to go here, do some imaging, then go here, do some imaging, then back here. So you can see, so we do have 3D imaging capabilities where you acquire a whole volume. Again, as Dylan mentioned, based on how many wavelengths you're using, each frame, multiply your frame rate by the number of wavelengths you're using, and then it goes to the next one. but it's an interesting idea, the ability to sort of go to maybe perhaps even different anatomy to see something like, biodistribution. I'm assuming that this person wants to get as close as they can to real time. I'd say we're not quite there yet, but the easiest way to do it would just be have, as few wavelengths as you can get away with, and then just do rapid subsequent 3D scans. You can actually set the step size, I think up to like 2 millimeters. So if you wanted to do a 3D scan that only had two slices, you could just repeat that scan over and over again, and you could do that pretty quickly.
Great, thank you. So we do have a bit more time. So if you, again, don't be shy. If you do have any questions, please feel free to put them in our Q&A panel. If we happen to not be able to get to your questions, rest assured, someone will be assigned to follow up with you and continue that conversation. So fire them off as fast and as much as you like. So the next, and that was a pun on photoacoustic firing, by the way. If anyone
Nice
knows
one.
me and who like puns. So the next question, when selecting a new molecular target for photoacoustic imaging, what is usually the key determinant for success? Target expression level, probe accumulation, or contrast agent properties? And in practice, are antibody-based probes often sufficient for moderately expressed targets, or do you typically need some form of signal amplification, such as nanoparticles or activatable probes? So I think the first we can answer that in several parts there. So what do you think is the biggest key determinant for success when evaluating novel contrast agents?
I think, yeah, I can start with this. So I think the most important is the compatibility of the contrast agent in generally. So keep in mind the wavelength that is fixed, and your contrast agent in generally should absorb in that specific wavelength. Then also, let's say in the case that you want to use this contrast in vivo, keep in mind the unmixing with other endogenous chromophores. So your contrast agent should give you a specific response.
This question is
Yeah.
an insightful one, and it's a very, very difficult question. I mean, you could apply this to any aspect of molecular imaging, really. If you've got a new molecular target, there are so many things that you need to characterize that have nothing to do with imaging. How well will your, antibody bind to it? How well is it expressed in what tissues? There's so much homework you got to do before you even get to the imaging part. But I think, Peter, I like the idea that you really want to select something, you know, you want to be able to select something that if you're interested in a new target, you want to target, that's your target. It doesn't really matter, is it highly expressed or not? It's your target of interest. So if it's not highly expressed, how do you image it? Well, then you're really selecting contrast agents that have very, very strong absorption in the wavelength range that we're using. That would be your best bet, is to have a very, very strong chromophore that's attached to, say, an antibody for whatever your target is. But that's a tough question. That's a big PhD project right there.
Yeah, and I think when speaking about, let's say, the PA amplitude, and it is maybe good to find what is the system sensitivity for that specific contrast agent or specific chromophore, starting in vitro imaging, let's say in some dedicated phantom. And then you should bring this concentration to in vivo project. But keep in mind always, let's say, the dilution of the contrast agent in the case that you are going to inject this contrast into the mouse. So you can calculate, let's say, an approximate volume of the blood, depending on the weight of the mouse. And you have to adjust more and more parameters, for the successful in vivo project. So I think to answer this question, it will be nice to get more details on specific applications, specific projects, and etc., etc.
Peter, you mentioned a phantom imaging with your contrast agent. Do you want to elaborate on that?
Yes, you can find, for example, in the system there is a library. You can find there the either the endogenous contrast, and I think there are two exogenous contrast. I think there is the spectrum of the ICG and some other contrast. But for you as a user, you can also save your own spectrum. So and then use this spectrum for further in vivo studies. So
Yeah.
this is something that the person who is performing the in vivo imaging should start with. So characterizing his her own contrast agent, save the spectrum, and then you can load the spectrum in multi-wavelength mode and use it for further in vivo imaging. Also keep in mind that when, for example, comparing the tissue, it's also nice to take, let's say, the spectrum before and after administration of the contrast agent. And you can also compare and see the changes that happen after the contrast agent injection.
Great. Right. So next question here, is there any data about using photoacoustic imaging in rats specifically? Lots of conversations here about mice, but what about rats? Our target is vascular damage and remodeling in the lungs in pulmonary hypertension. So
Yeah,
comments
so
about what's possible in rats.
yeah, as far as rats go, absolutely possible. Most of my PhD work was in pregnant rats. So that is, definitely a model that we can work with photoacoustics. As far as imaging pulmonary hypertension, it starts to get a little tricky just because you have, the ribs that are going to interfere, and also the lungs, are filled with air. So it's impossible to get an ultrasound signal through air because there's no transfer media. So have been some publications, showing different ways to work around that and some different parameters that can be measured. But it is, not quite as straightforward as, imaging a liver.
Yeah, I'd be kind of curious about the application there. For example, if you've got edema in the lungs, there's your way of conducting ultrasound. Yeah, that's an interesting model. I haven't heard that one before, but that's the depth isn't so much the problem, I guess, Dylan. It's more just about
Right.
imaging through air where the ultrasound doesn't penetrate.
Yeah.
Absolutely. Okay, so if we don't see any other questions come in, maybe we'll do one last question, and you still have a chance while they answer this one to put in yours. So this is about oxyhemo, oxyhemo and deoxyhemo. They are curves that are available in the Vevo library. If they're theoretical curves, is there a difference from reference curves in Vevo? Does the difference affect unmixing? So I'm not sure if this one is referring to if those curves have been validated. Maybe someone can comment on the validation of the oxy-deoxy curves.
Those curves are, let's say, the standardized spectra of the either oxyhemoglobin or deoxyhemoglobin. So the system is using those two curves to unmix those two compounds. And then, for example, when scanning, especially in the multi-wavelength mode, and you want to put the third or the fourth chromophore compound, then the system is adding more wavelengths. Then you may maybe have slightly different wavelengths in the case that there is an overlap, let's say in between, in between, let's say, oxyhemo and your exogenous contrast agent. So then the system can slightly change the wavelength, and also is putting more wavelengths to be able to distinguish between two or more different chromophores. So the more chromophores you want to image, the more wavelengths you have to scan.
I'll add here as well. I think theoretical may be coming from what I showed in the early slides there about the collagen curve being labeled as theoretical in our library. So that one's labeled as theoretical because there are so many different collagen variants, and it's been observed in the literature that those different collagen variants have slightly different spectral curves. As far as oxy-deoxyhemo, they're not labeled as theoretical in our library, and those curves and the data from our system has been validated against technologies like bold MRI and several different models of disease, over the course of many different years and different publications. And we can certainly share those if you're interested. Okay. Does anyone want to, from our panel here, want to add any? Well, maybe we have one more question that snuck in. Let me see. Oh yeah, let's do this one final question. It's also about rats, so we stayed on that theme. So last question, but if you do have any more, fire them off, and then oh, there are a couple more coming in. Okay. So I think we'll make our experts hold off for a few more minutes here so we can answer these questions. We are studying fetal rat lung development after treatment of pregnant dams with nitrophen. Can the software measure lung volume after a 3D scan? Can the photoacoustic imaging be overlaid on a 3D scan?
This is in your wheelhouse, Dylan.
Yeah, so I
Defer to
mean,
Dylan here.
yeah, off the bat, yes, the PA can definitely be overlaid on the B mode for 2D and 3D. So that is definitely doable. As far as measuring lung volume after a 3D scan, absolutely. I would probably rely more on the your higher frequency transducers to get that resolution. It is, again, tricky. Obviously the fetal lungs are not full of air, but they are, it takes a little bit to, differentiate and become familiar with the anatomy. But that is definitely feasible with a 3D scan of, you can do whole abdomen, individual dams, whatever your interest is.
Sounds like an interesting study.
Yeah.
Mhm. Can I
Thanks.
ask a question, Kristiina?
No. No,
Well, we have our application scientists here. They're experts in the field and they're traveling around. They see many customers. They're, training them. A question for them is, what's the most interesting photoacoustic application they've seen in the field?
I mean, in general, each application is really interesting, and especially when you are seeing new applications, new things. Personally, I like the oncology, so imaging of different tumors, especially the orthotopic tumor, that will gives you, yeah, you can see really differences in microenvironment and also the impact of the drug treatment, and yeah, that's really nice.
Yeah, I agree. I think all of the trainings and customers I've interacted with, there's like a nuance to everything that it makes it different and interesting. So kind of keeps you invested in their study and then following up, reading the papers when they come out. But I think for me, the contrast agent development is really kind of it's definitely a hot field, and it's something that's really cool. All the different ideas that people are coming up with, whether it's like metallic nanoparticles or organic dyes, and how they're kind of modifying them and targeting them to different markers for a range of applications.
I could add into that what I find fascinating, as well as like the nanoparticle development, and kind of in conjunction with that is the development of novel targeted therapeutics and photothermal agents, nanomedicines, activatable agents. It's a really cool field, in my opinion. And yeah, it's really interesting to watch it grow, and obviously with the main goal of providing things like targeted treatments for cancer patients, for instance, is quite an interesting endeavor, and hopefully that will continue to develop. Drew, did you have something in mind you wanted to share as well?
Oh yeah, I mean, I was doing, sort of trainings and field-based work for quite a number of years. I mean, I have a neuroscience background, so the brain is kind of my favorite thing to study from a science point of view. But yeah, just looking at, a new way of doing functional brain imaging, I would say is just really interesting. The neuroscience field doesn't use a whole lot of imaging in the preclinical space, and when they do, it's often microscopy. It's often invasive. In some cases, people have access to a magnet and they can do some MRI imaging. But this really, I feel like it really opens up that neuroscience field to be able to look at real-time oxygen saturation in the brain as a measure of brain function. It's a perfect analogy for functional MRI. The only difference is it's way more sensitive. And so that I think there's some really, really kind of cool stuff going on there. One of the more interesting applications I saw was lymphatic pumping in a mouse. So you inject a dye, indocyanine in this case, in between the toes of a mouse, and it'll get drawn up into the lymphatic system. And they were able to measure the rate of the pumping. The lymphatics are actually muscular; they actually pump, and they were able to measure the rate of pumping of lymphatics in a mouse. Now you can imagine, lymphatics in humans, we're talking, sub-millimeter structures. You can imagine in a mouse, even with our ultra high resolution, it's really, really hard to see, if not impossible in B mode, but you could actually see it with photoacoustic imaging. So just being able to do stuff that you just couldn't do before, not just with our technology, but sort of in science, and explore questions that you've never been able to explore before. That's the kind of neat part.
Great, thanks for sharing. So we're at the 2 more minutes till the top of the hour. So as I pull up a slide that tells you all the different ways you can connect with us, I'll pose a final question to our panel here. So for unmixing, is it better to acquire an image at more wavelength points? So basically, how do you know how many wavelengths to include in your multi-wavelength scan? Is it better to add more kind of in general?
I think the best you can do is just load your spectrum directly into the multi-wavelength mode, and then system will automatically detect or decide what are the best wavelengths to unmix all the components.
Great. All right. Well, thank you so much to our audience and all the engagement. We really appreciate connecting with you in any capacity. If you'd like to connect with us some more, please visit our website. We have an excellent resource portal for our customers with lots of different resources there. You can submit any questions or contact us using our web forms on our website. You can register for upcoming webinars. Find us on our social media channels. We're quite active on social media. And of course, you can visit us, and we'd love to see you in person at any conferences you're attending. So thank you to our panel, very knowledgeable, experienced photoacoustic imagers here. So thanks for all your responses, and thank you again for everyone who joined, and have a lovely day.